All right, this is part 2 of chapter 28, continuing along discussing green algae, land plants, introduction. So we wrapped up in the last lecture, finishing up talking about the function of stomata and the waxy cuticle. If we look at leaves, the guard cells that open and close that stomata, allowing CO2 to come in, oxygen to leave, but also controlling water loss. It's a very important adaptation. So in addition to the adaptation of cuticle and the stomata, these land plants had to get the water up from the soil up through the entire length of the plant. So they've really evolved to have a vascular system. And this vascular tissue, these specialized cells are just basically long cells, vessel-shaped cells called tracheids, pretty well-evolved structures. And the land plants, that vascular plants, the gymnosperms, the angiosperms, the complex plants are going to have these tracheids, and really, we'll see those very prevalent in these highly evolved plants. And what makes them different is, in addition to the cell wall that's around all plant cells made of cellulose, these specialized cells also have a secondary cell wall that's much thicker, and it's lined with lignin, and that allows for water to flow really efficiently through this thing, like a tube. There's pits on the ends of the cells that allow water to flow efficiently between the neighboring tracheids. All right, so these are going to be, again, very specialized, but not as specialized as vessel elements, which we'll see in a second are the most specialized type of water-conducting cells. They're shorter and wider, and they have gaps in both ends of them where the cell walls are missing. So again, if we take a look, the earliest cells to be on land had to have some kind of, or the earliest plants to be on land had to have some kind of cell that was going to move water, right? So they had this very simple primary cell wall with cellulose, no secondary cell wall yet. We find this in fossils of old plants and also in the very simple present-day mosses. Years later, millions of years later, we start to see the first vascular tissue becoming more complex. So now we've got some structural support in this, and we can better move the water. All right, the increased structural support that's found in all tracheal or in all vascular plants are these tracheids, and these have pits where now water can move from cell to cell, whereas here we didn't have that. Okay, we didn't have that in these initial ones. And the most complex found in the neophytes and the angiosperms, which are the newest, I guess, evolutionarily plants, have perforations in them. So we really get an efficient transfer of water through these cells, water going up through these vessel elements. Okay, so that's how water gets there on land. But looking at reproduction in dry conditions. So in eukaryotes, the life cycles involve very different variability structure than do animals, for example, right? So what we see is we still see the same kind of processes. We still see mitosis, we still see meiosis, we still see fertilization taking place with plants, but the players are different, and the way in which these processes go is very different than in animals. So in animals, you get you got all of your cells, and they undergo mitosis, and then some of those cells undergo meiosis to become egg and sperm. Egg and sperm come together through fertilization, and then you get a zygote, and the zygote undergoes mitosis, and you get a person, and the process continues. Now, it's a little bit different because in plants, we're going to see here that we get an alternation of generations, which is very unique to from us, right? But we did see this already in protists, and I told you it was going to be coming again. So I hope you spent some time with that when we did protists the other week. So 3 innovations were very important for plants in terms of reproducing in a dry environment. So they need to have spores that were going to resist drying, because obviously the spores now are no longer going to be in the water where they're where drying out is not an issue. Now they're going to be out of the water. So to resist that drying, they're encased in a tough coat of something called sporopollenin, and allows them to be transported long distances. Right? We're also going to see the gametes are protected by structures called gametograngia that were producing complex multicellular structures. And we're going to see that embryos in angiosperms are going to be retained and nourished by the parents here. So again, much more advanced than just simply releasing the spores and hoping they end up finding their counterpart out there to fertilize. So we're looking at these adaptations being crucial for survival on dry land. All right. So again, one such example here being the complex protective reproductive organs. So individual plants produce here distinctive male and female gametogangia. All right, so this is important because we're going to see kind of the distinction here. So sperm-producing structures are called the antheridium or antheridia, and the archegonium or archegonia produce eggs. Okay, so these are analogous to the testis and the ovaries of animals. So the sperm-producing structures, the antheridium being the testis, and the archegonium being like the ovaries in animals. And we will see these terms coming up, ovaries, later on in plants as well. So try not to get that too confused, but just to give you that analogy. So here's the male structure containing the sperm, the antheridia, and the female structure, the archegonia, containing the eggs. Okay, and moss gametes are pretty distinctly either going to be male or female. Okay, and you can kind of pull out and see what those look like. And you've seen mosses before, you could probably identify them if you did look at them. And those of you that may have taken the lab course before will take the lab with this lecture class. You'll take a look at those in lab. So instead of shedding their eggs into the water or soil, land plants retain their eggs and they form inside of the archegonia. And that prevents them from desiccation, drying out. And also, it's, you think about it, it's more controlled. All right, it's more controlled, it's more advanced. In contrast to most green algae, zygotes, or the union of the egg and sperm, begin development in the parent plant, and they form multicellular embryos that remain attached to and can be nourished by the parent plant. So lots of things here that are, going to be important for the plants to survive on land. All right, so I mentioned alternation of generations as being the life cycle here of plants. So in conjugating algae, stoneworts, collocates, the multicellular form is haploid, and the only diploid form is the zygote. But we're going to see that there's other examples of this. So the multicellular haploid phase in the alternation of generations cycle, which we're going to go over, is called the gametophyte. Basically, what this is in this cycle, it's a giant egg cell or a giant sperm cell, the gametophyte. The multicellular diploid phase is the sporophyte. So haploid means half the number of chromosomes, and diploid means the full number. And these two are going to alternate back and forth between the gametophyte phase and the sporophyte phase. And we're going to see that alternation of generations takes place in between the two. And sometimes the gametophyte dominates the cycle, and sometimes the sporophyte dominates, just depending on the type of plant. So for all of these cycles we're going to look at, there's the same sequence of events. Okay, so you start with the sporophyte, probably the easiest thing to do, and the sporophyte produces haploid spores by meiosis. Okay, that's the first step. Spores then germinate and divide by mitosis and develop into a multicellular haploid gametophyte. Now that's bizarre. Again, this is like basically a sperm cell by itself or an egg cell by itself undergoing mitosis and just becoming giant. And that's essentially what happens here. And in the more advanced plants, it doesn't get that big. But in the early plants, the gametophyte does kind of dominate. From there, the gametophyte serves its purpose and produces unicellular haploid gametes by mitosis. So those two gametes that are then formed from the gametophytes are going to unite, form a diploid zygote. Okay. The zygote then undergoes mitosis, similarly to what we see in animals, and we end up with the sporophyte, which is the 2N, the diploid plant. Okay. So here's a very simple look at the alternation of generations for just a basic land plant. So here's your sporophyte. Notice it's multicellular, it's diploid. It undergoes meiosis to produce sex cells, which are spores. Okay, those individual spores then undergo mitosis themselves, and they get bigger and they turn into this gametophyte. From there, the gametophyte produces its own gametes, okay, and those gametes will come together via fertilization to produce a zygote that will then undergo mitosis to become a sporophyte. Okay. So this slide is just describing what is happening. Okay. So let's keep some of these terms straight just so that we're all on the same page. So zygotes and spores are both single cells that divide by mitosis to form a multicellular individual. Zygotes develop into sporophytes. Okay, so here's our zygote developing into the sporophyte, the 2N. Spores develop into gametophytes. So spores are haploid, gametophytes are haploid. Okay. Zygotes result from the fusion of two haploid cells, such as sperm and egg, but spores are not formed by the fusion of gametes. All right, spores come from meiosis of the sporophyte. So spores come from a sporophyte. It makes sense. Spores are produced by meiosis inside structures called sporangia. Gametes are produced by mitosis inside of the gametogangia. Okay, so spores, sporangia, gametes, gametogangia. So keep those terms straight in your head, and they do kind of go together. So when we're talking about those non-vascular plants, your mosses and your really early on plants, the sporophyte is small, short-lived, largely dependent on the gametophyte for nutrition. So this is where we see a gametophyte dominant life cycle. So in mosses, and you'll see these pictures in your book or in the mastering, you'll see that in mosses, the gametophyte is the green part, and then the sporophyte are the little yellow spikes kind of coming off of mosses. When we get to more complex plants, ferns, your gymnosperms, your angiosperms, these guys, the sporophyte is much larger and longer lived than the gametophyte. So we call that a sporophyte dominant life cycle. Now in ferns, you can look and you can find the gametophyte if you look really closely underneath. It's very tiny. But in gymnosperms and angiosperms, your pine trees, your flowering plants, the gametophyte, we're going to see in a second, is microscopic. Okay. So here we're looking at a gametophyte dominant life cycle. So this could be a moss. And notice that the gametophyte is large in the mature plant. And this is a gametophyte. This is a gametophyte as well. And then the sporophyte is up here at the top. So if we look and we just kind of follow the sporophyte, you can see the sporophyte is producing spores. We know spores are dispersed by wind. And those spores will then undergo mitosis to become large gametophytes, which is what's happening here. Okay, and the gametophyte is doing pretty well on its own. Here's a mature female gametophyte. Okay, and if it's kind of left to its own devices, it'll develop into that female gametophyte. If we're looking at from the gametophyte sperm being produced that swim into the egg, we get a male gametophyte here. Okay. So in our female gametophyte, you can kind of zoom in here and you can see that the eggs are going to develop in the archegonia, which we talked about earlier, and then those eggs will fertilize, and we end up with a zygote, okay? And the sporophyte will develop. Remember, the sporophyte has to be produced via fertilization. So you have to have the union of two sex cells in order to get a sporophyte. So here it is developing, and then along the outside here is going to be your gametophyte, and the sporophyte's developing in the center, and that's kind of where they come together. All right, so an interesting life cycle here for the gametophyte dominant life cycle. Much easier, I think, the sporophyte dominant. This is more kind of common what we see. So here's our mature sporophyte. It's producing spores in sporangia. Those spores are dispersed by wind wherever they land. Conditions are right, they'll develop into a gametophyte. Here's our mature gametophyte. The gametophyte produces sperm. It produces eggs. These things come together. Fertilization takes place. Okay, and this can happen, right here, right next to the gametophyte. And you can see the gametophyte is still there, but our sporophyte is starting to grow here from this zygote, and the sporophyte is dominant, and the gametophyte is very small. Okay, but it is there, and you can generally find that right next to a fern. So there's two ways in which we look at plants producing spores. So there's heterospory and there's homosporous. Heterosporous or heterospory means hetero, that prefix means two different or two distinct types. Homo means the same. So if we have two distinct types of spores produced by different structures, it's heterospory. And then in terms of non-vascular plants and seedless vascular plants, they produce a single type of spore that's going to develop into a bisexual gametophyte that's going to produce both eggs and sperm. Okay, so this is going to be much more simple. So we've got our sporangium, which produces spores. Spores produce the gametophyte, and then the gametophyte produces both the eggs and the sperm, which is what we're seeing right here, the gametophyte producing both the eggs and the sperm. Now, in heterospory, we see a different situation here, and so seed plants are going to be more complex. So now we see something called, the microsporangia and the megasporangia. So in heterosporous species that are more complex, we've got separate structures here, the microsporangia and the megasporangia. So the microsporangia is ultimately going to produce sperm. So microsporangia produces microspores, which produces a separate male gametophyte, which makes sperm. The megasporangia produces megaspores, which produce the female gametophyte, which produces eggs. Okay, the gametophytes of the seed plants are either male or female, but never both. So we have two separate gametophytes. So here we just had one gametophyte. Here we're going to have two gametophytes. I'll show you this in a second. All right, so this is more complex. So you can, think about different ways to remember this. Mega, female, I don't know, something maybe you can come up with to remember the differences there between the two. All right, so another structure we're going to see here is pollen. And pollen is another term for the sperm of non-vascular and seedless vascular plants. Okay, and these have to swim to the egg to fertilize it. All right, water must be available for fertilization to occur in this case, right? So yes, that's sperm for non-vascular seedless vascular plants is analogous to the pollen grain of land plants. All right, so pollen grain in land plants allowed for plants living in dry habitats to reproduce efficiently. Okay, so we're going to see kind of the function of that here in a second, but tiny male gametophytes surrounded by a tough coat of sporopollenin makes up a pollen grain. So it is a tiny male gametophyte, and it's surrounded by, again, this structure that's nourishing it. Can be exposed to the air for long periods of time without drying out, and can be carried to the female gametophyte by wind or animals. So again, this leads to lots more possible variations and lots more ways in which pollen being very important. And when pollen evolved, these heterosporous plants no longer need water to accomplish fertilization. All right, and that's very important. So instead of swimming to the egg like the naked sperm, did before, the tiny male gametophyte can now, take to the atmosphere of the skies, to animals, right, to fertilize, and that opens up a lot of evolutionary doors. So let's look at a seed. So seeds are really important. So a seed includes an embryo and a food supply surrounded by a tough protective coat. So the seeds enable embryos now to be dispersed to new habitats, right? So here is our embryo. Here's a layer of nutritive tissue, and then here's the protective coat in the seeds. So seeds are great. And we can see kind of a better look here, the traits of seed plants when we look at the life cycle of a pine tree. So we are going to spend a little bit of time on this because it is kind of important. So I'm going to walk through the steps here, and then I'll show you the picture. So in the life cycle of a pine tree, it's typical heterospory in a gymnosperm. So remember your gymnosperms being like your obviously your pine trees and your non-flowering vascular plants. So if we start with the sporophyte, which you'll see in a second in the left, there's two different structures called cones. There's separate structures called cones, which develop into the microsporangia and the megasporangia, which we talked about, the micro making the male counterparts and the mega making the female. The microsporangia contain a microspore mother cell that divide by meiosis to form microspores, which then divide to form pollen grains, which again is the sperm. Megasporangia found inside protective structures called ovules. Megasporangia contains a megaspore mother cell. Okay, that divides by meiosis to form a megaspore. And all of these things are ultimately leading to the function, or to the egg and the sperm being produced. The megaspore in the female part is going to undergo mitosis to form the female gametophyte, which produces egg cells through further rounds of mitosis. The female gametophyte stays attached to the sporophyte as pollen grains arrive and produce sperm that fertilize the eggs. Seeds mature as the embryo develops. Inside the seed, cells derived from the female gametophyte become packed with nutrients provided by the sporophyte. Okay, so let's walk kind of through this and look at the different structures. So here is our mature sporophyte. This is a trait. It's 2N, it's diploid, it's got the full number of chromosomes. It's going to produce two different types of cones, the cones with the microsporangia and the ovulate cones with the megasporangia. So let's look at what's happening here with the male side, and we've got the cones with their microsporangia producing the microspore, which is ultimately going to form the pollen. All right, pause there for a second. Those pollen grains disperse via the wind. And I'm sure you've all seen cones, pine cones. Now you know what they are. They're the reproductive structures of this pine tree. So we've got our ovulate cone, the female one, which contains the ovules, which contain megasporangia, which are going to become the megasporangium, which have the mother cells. Okay, these mother cells are going to undergo meiosis, and what they're going to do is they're going to produce this, this egg that's going to this megaspore, this egg cell that's going to be fertilized by ultimately by the pollen. So the megaspore divides to the female gametophyte, okay, in this case. And you can see the female gametophyte is within this tiny structure, right? So like we said before, it is microscopic. So megaspore divides to form the female gametophyte, which forms eggs by mitosis. Okay, only one of those eggs is fertilized and will ultimately develop into an embryo. Okay. And that embryo will then be dispersed in the form of seeds, and then those seeds will ultimately land on a place where they are going to be in the right conditions to grow. So they can stay for a really long time before they find that condition. All right, so take a look at this. There's a good, in your mastering this week, you'll see there's some good activities that'll help you to reinforce this stuff. It can be a little bit tricky. Okay, so do make sure you read up on this in your book as well as listening to the lecture. Okay, so we're almost through here. So you thought this was a little crazy. Flowers are also crazy, but not too bad. So flowering plants, or the angiosperms, most diverse, 350,000 species. They've totally taken over in terms of, in most places, as the dominant species of plant on Earth, on land. And the success of these flowering plants, you could probably guess, has to do with the flower. And if we look at flowers, you see there's a structure in there called the stamen, which contains the anther, where the microsporangia, or the pollen, is going to develop, and the carpel, which contains the ovary, where the ovules are found, and that's where you're going to find the megasporangia, which will develop into the female gametophyte. Now, you guys have probably dissected a flower at some point in your, I hope, in your schooling career, in maybe 7th grade or 8th grade or something like that. So you're probably familiar with what a flower looks like. Lilies are usually used to dissect. Show you a picture here coming up. One of the things that's really interesting and really cool that happens with these flowering plants is that you actually end up with two sperm cells fusing with two different female gametophyte parts. So one is going to fuse with the egg to form a diploid zygote. And that makes sense, right? That's the one that's going to develop into your sporophyte and go on to be your flower. But then there's another sperm. So another sperm gets in there, and it fuses with two nuclei in the female gametophyte. And it forms something called a triploid 3N. So we've got triple the number of chromosomes, but it doesn't develop into a flower. It develops into nutritive tissue called an endosperm. This ultimately will become fruit, like the fleshy part of fruit. So really pretty cool. Take a look at this. So again, a little bit more going on here, but fairly simple. So you've got your flower, and there's the stamen here, and this is the male part. And the stamen is, if you look, focus on top of it, the anther. Meiosis takes place in the anther to form microspores. Microspores become pollen, the male gametophyte, and then the pollen is going to be dispersed. So where does it go? Well, it can either go back to the flower itself, or it can go to other neighboring flowers. So if we zoom in on the bottom of the carpel, this is the female part where we find the megasporangium, and the megaspore is in there, retaining the ovary. We got our female gametophyte in there, developed microscopically, and we got eggs in here. So when the pollen goes in, it travels in the egg and it can fertilize, or the pollen can get in and it can fertilize the eggs there down in the carpel. But remember, it's not just one sperm. There's a double fertilization that takes place. So in addition to one egg being fertilized, the nutritive tissue forms when two more cells get, or one cell with two nuclei gets penetrated. And the nutritive fruit develops in the ovary wall from that endosperm surrounding the seed. And depending on the type of flower, it can be a lot or not a lot. I'm thinking about things like an apple and a peach and stuff like that. There's a good amount of that endosperm that's produced. Once this finds a spot via wind, animals, whatever, it'll latch on to an area and it'll develop into a new flower, new sporophyte. So I think this is a little bit easier to understand than the previous life cycle, but you should get the idea here of what's happening with these angiosperms. So this is the most complex and this is the most adaptive value. So a couple of specific things that happen. So after they evolved, stamens and carpels became enclosed by these modified leaves called sepals and petals. So natural selection, forged this change here to have these colored or different smelling flowers. So carrion flowers smell like rotting flesh and attract flies, so then they can take the pollen different places. Hummingbird pollinated flowers, red long tubes with nectar at the base, right? These are adapted to coevolution with the hummingbirds. And then bee pollinated, one of the most common ways we see pollination and one of the most effective is with bees. So we need bees, and they are attracted to bright colors, bright purples, things like that. Right, so we talked about fruit a little bit, but fruit's a structure that is derived from the ovary and encloses one or more seeds. Okay, often nutritious, like we said, that endospore it's nutritious to the developing ovary, and it's nutritious to you to eat as well. While the evolution of flowers made efficient pollination possible, the evolution of fruit made efficient seed dispersal possible. So the apple falls off the tree and it lands, and it's good to stay there throughout until the season is right. So adaptive radiation just means that a large number of species are dominating an area. So in order for this to take place, a species has to be or a lineage, I should say, of organisms has to be very well adapted to a variety of habitats because it's happening all over. So angiosperms, these flowering plants, represent one of the greatest adaptive radiations in the history of life on Earth. So multiple sort of evolutionary characteristics allowed for this, specifically those water-conducting vessels that we talked about, the flowers themselves, and fruits. So all of these things that we've been discussing really lead to, this idea that the angiosperms are the most dominant of the flowers, and that's why, or the most dominant of the plants on Earth, and that's why we see them around in most areas. So if we look at morphological traits and things that we can observe on the angiosperms, we divided them into two major groups, so the monocots, or the monocotyledons, and the dicots, or the dicotyledons. And this has to do with differences at the microscopic level with the cotyledons, or the first leaves that are produced after these things start to grow. So cotyledons store nutrients, provide them to the embryo. So in monocot species of angiosperms, they only have one cotyledon, and dicots in this early stage have two cotyledons. So that name mono, meaning one, cotyledon. So here's your cotyledon, this first leaf, and then your dicots have two cotyledons. So this is how you can tell them in an embryo. You can look at the vascular tissue, and you can see that in your cotyledon plants, you've got the vascular tissue totally scattered throughout, right? So these are your tracheids and your vessel elements and things like that. But in the dicots, the vascular tissue is arranged around the outside. You can also tell the difference between a monocot and a dicot by the vein structure in the leaves. So monocots, like grasses are monocots, they have parallel veins in their leaves. And also in flowers, they have petals in multiples of 3. In your dicots, you've got the branching pattern in leaves, and their flowers are in multiples of 4 or 5. Okay, so that's an easy way to tell the difference. Is it a monocot? Is it a dicot? Again, those are morphological characteristics that are observable. All right, so the key lineages or the major types here that we look at, just kind of summarize the green algae, like we said. Traits that are common to all green algae, they have, double membrane chloroplasts that have chlorophyll A and B. We'll talk about that more when we talk about photosynthesis, but they're important producers near the oceans, in the shores, freshwater environments, and we see things that, have symbiotic relationships. We talked about like lichens, freshwater protists, right? Very important green algae in that respect, right? So the one that you want to recognize are these olvophytes. And these olvophytes, they're marine, they're freshwater. They reproduce both sexually and asexually, and they have a dormant winter stage. Many are diploid as zygotes, although some multicellular species do have that alternation of generations that we talked about. In terms of these guys and their importance or their relevance, they're primary producers in freshwater and coastal marine environments. We also use them for types of lettuce and stuff like that. Okay. So in terms of the non-vascular plants that you want to recognize, so non-vascular plants, the first lineage to break off of the land plants. In these non-vascular, these simple ones like the mosses, remember the gametophyte is dominant, longer lived phase. These mosses are anchored to rocks, soils, tree bark by rhizoids. They lack vascular tissue. They don't have that lignin reinforced cell walls. They don't have those tracheids or those elements in there. They have to be near water because they have flagellated sperm that swim to the eggs, and their spores are dispersed by wind. All right, so you want to recognize the mosses or the bryophytes in this case, and the bryophytes being just mosses. Simple conducting tissues in this group, no lignin. They often dry out. Okay, they can become dormant as well, and they can withstand that drying that way. Probably seen this with peat moss as well as a different type of peat moss. Stores a lot of carbon. Peat is widely used in gardening and soils and things like that. The seedless vascular plants, these are a paraphyletic group, right? They branched off many times. Vascular tissue does have those lignin reinforced cells. The sporophyte here is dominant. It's a longer lived phase of the life cycle. But we do have a gametophyte that is physically independent. So that is different from the seeded vascular plants, the angiosperms. The eggs retained on the gametophyte and sperm swim to egg with the flagella. Sporophytes develop on a gametophyte and are nourished by the gametophyte and small. Okay, so these are your club mosses, your ground cedar. All right, these lycophytes, which we're going to say, small plants live on the forest floor of tropical trees. They have roots. They have really unusual leaves, as you can see, and they do live symbiotically a lot of times with fungi. In terms of the human impacts and relevance, right, these were really important during the Carboniferous period, the coal-generating forests. Whisk ferns. You've probably seen these maybe about, there's only about 6 species left, and they're all tropical. Maybe you haven't seen these actually. But these, you want to recognize what these look like. They gain nutrients from fungi, and they are seen sometimes in landscaping. But I was thinking of something else. I guess we're not really, unless any of you are from Japan, you've probably never seen these, but I feel like I have seen them around. Ferns, I'm sure you've seen. Ferns being a seedless vascular plant. Again, vascular having that vascularity. Seedless, no seed. So very common here. Some people eat these, believe it or not. Culinary delicacy, and they're widely used in people's houses as ornamental plants. Horsetails, again, you've probably seen these as well out kind of by a pond or something like that. They prefer wet habitats. They allow oxygen to move down the roots. They reproduce sexually and asexually. They have a sporophyte dominant life cycle, and the gametophytes are small and short-lived. So, like I said, you've probably seen these horsetails around before. All right. Lastly, the gymnosperms and the angiosperms, kind of putting those together here. You want to recognize some of the gymnosperms and recognize that these are your seed plants. They're monophyletic. And they're really defined by their synaptomorphic traits of producing seeds and pollen grains. Angiosperms produce seeds and ovaries. Gymnosperms do not. That being a major difference there. Let's see recognize your ginkgos, ornamental plants, your cycads, sagopalms, right? and your pine trees as being examples of gymnosperms. And then all of your flowering plants are angiosperms. Okay. Neophytes, also gymnosperms as well. Okay. That will end chapter 28. Thank you for your attention.